Thank you. So I'm going to talk a little bit about a project I've been working on over the last 10 years or so. It's how to use our DNA sequencing technologies, particularly for getting relatedness estimates. So why would we use sequencing for genotyping? And one of the reasons is that it's got minimal setup costs compared to alternatives. We can use some common equipment that we use for other reasons, common equipment, materials and protocols. There are lab methods that allow us to assay just a small proportion of the genome, so that reduces the costs. And we can get adequate information even from a low depth sequencing. I'll explain that a little bit more later. So the driver here is we want to get cheap genotyping tests particularly for our primary industries. The downside of all this is that it's messy data, so it requires some technical bioinformatic and statistical approaches to deal with it. How do we get these genetic variances from the sequencing reads? So we filter the DNA to a subset of the genome, as I talked about earlier. We then look for sequence differences at a particular position, and the differences we'll call A and B. And then the sequencing reads that we get translated into counts for each individual that gets sequenced, for each genomic position that passes through that filter, and then of each particular variant, the A and Bs. So we end up with these count matrices. And a diploid individual, such as humans, they can have three true types, so AA, AB, and BB at a particular position. So if we concentrate now just at one individual in one position, and suppose that individual is truly a heterozygote, so it's an AB type, then we've got different possibilities for what we see from the different reads. Can I point with this? No. Yeah? Not very well. Okay. I'll try and describe it. So in this table, in each row, we've got a particular number of reads we've seen at that position for that individual. And if we only have one read, then we only see an A or a B with equal probability. As the number of reads increases, there's more chances that we see both of the true variants that are there, so both A's and B's, but there's still a chance that we'll only see one of the types, only A's or B's. Now, within a row, those numbers add up to 1, and it's the A's and the B's are the same, so there's really only one thing we need to look at. And so I'll concentrate on this probability of only seeing A's at a given depth for a true heterozygote. And I've called this capital K, and that gets used in these equations to work out the relatedness. So if you look at a relatedness estimate, a common one, I've looked at that using these probabilities and going through to see what do those relatedness estimates look like given this low depth sampling. The first thing to cover is that not every individual has reads at every position, and so we need to deal with the missingness. And I simply take away anything that doesn't have a read, I take it away for each pair of individuals I'm looking at. So you only end up using the ones that are seen in both. And then if you go through the mathematics of those equations, we find that the inbreeding estimates that we'd normally get, that equation depends on this value of K. And so there has to be an adjustment made so we can get back to the inbreeding that we're trying to estimate. And for a pair of individuals, if you go through those equations as well, we find out that the long-term average of that doesn't depend on this value of K, and so we don't need to make any adjustment. We can just use those formula as it was given. So to do this for many individuals, it's got a lot of computational power, And to make that simpler, there needs to be some methods to cope with the fact that we've got these different numbers of positions, different variants, and different sets of individuals. And so I've developed methods to do that, and I've made software available online. Right, that was the hard bit. Why focus on relatedness estimation? So it's got lots of uses. Identity checking, do these two samples come from the same individual? Parentage checking, is our parentage records right? Or more commonly in a lot of these fields, parentage discovery, who is the parent of this individual? Estimation of genetic merit, something that's called genomic selection, particularly in our livestock industries, is sort of the modern way of approaching the evaluation of the genetics of individuals. Estimation of genetic parameters such as heritability, and studies of the genetic structure in a population. And by doing that through this relationship estimation method, we avoid these issues of the missingness in the data that become a problem with other methods. So some examples of this working in practice. This is it's actually an Australian goat breeder, very large operation. They came to us looking for help with their breeding program. That had they hadn't been able to do it very well until we had our sequencing-based approach made it affordable for them. They started with us in 2017, and you can see from this plot that they've made great genetic progress since they started with us. And then a population genetics example, and Neil's might be here somewhere. But so this was work with University of Otago. Some fur seals were sampled around mostly the South Island and the lower North Island and bycatch. And you can see here with these colours that the statistical representation of the genetics of those samples mirrors where they were found mostly. So the West Coast ones are on the left and the East Coast ones are on the right, for example. The exception there is the ones in light blue that are at Kaikoura. That was a new colony that was having lots of immigration to that point, and so you can see that the light blue dots do spread across that plot. And I finally just wanted to show you, so this method we call genotyping by sequencing, and here's a plot of the number of samples that we've genotyped in our lab at Invame. You can see there's a great diversity in species, some of them very many and some of them not so many, but many different species and for many different uses, both primary production and ecological studies. So I also want to use this diversity slide to acknowledge the diverse range of people that have helped me in my career, my mentors, my colleagues, those that have got funding, and my family. And lastly, thank you for the Royal Society Te Aparangi for this honour. Thank you.
Thank you very much for that elegant explanation. I'm just curious as to how you went about developing the mathematical aspect of it. Was that years of work, or was it something that really came together quite quickly?
It was more than 1 year. And with genetics, there's always a 2 that gets you with these, because of the diploid nature of things. And often when you find it looks really bad, you go back into the equation and you find a 2 that was in the wrong place or not there, and it comes out nicely in the end. But yeah, it did take a while. So we aim for about 2 to 3 reads per individual per site. So that is very low from what people would normally go for.
So does that mean you can scale it up to hundreds of individuals?
hundreds, thousands. I mean, the deer there were over 100,000 we've done. Yeah, yeah, exactly. Yes. Yes.
Did you come to it from a mathematical perspective or a genetics perspective originally? Or a bit of both?
My colleagues asked me, we can't afford to do the deep sequencing. Can you make this work with, shallow sequencing? So, yeah, exactly. Yeah.